In part 1, you have learned that in order to reverse glycolysis to produce a glucose molecule, there will be 3 reactions that become energetically unfavorable because they will have a very positive delta G value. And our focus here now is to study how does liver make all these reactions possible. And let's look at the first one. Well, the delta G of the conversion from pyruvate into PEP will be so positive that we couldn't do this in one step. Instead, we have to do it in 2 steps, and each one will require a large amount of energy investment in the terms of ATP and GDP. And for the remaining 2 steps, the reversal cannot be done by using kinases because the energy cost of putting a phosphate group into the ADP to form an ATP is so big that it is impossible to be done. Instead, our cell will employ enzyme phosphatase to reverse the reaction. So what phosphatase does is to cleave the substrate and to release the phosphate group along with the energy that will power the reaction. And so over here you have F16PP forming F6P. So it is the cleavage of that phosphate group that provides the energy for this reaction. And likewise, it is the cleavage of this phosphate group from G6P that will power up this reaction. And for both reactions, you can see how water molecule is involved because it is a hydrolysis reaction. Okay, I just want to show you the free energy values for these 3 reactions. Going down in glycolysis from PEP into pyruvate, you have a delta G of negative 17 kJ per mole. So the reverse process will be positive 17 kJ per mole. And can the reverse reaction in gluconeogenesis happen spontaneously? Well, no, because it's positive 17 kJ per mole. And therefore, we will require a 2-step solution by which a GDP and an ATP are invested. And the same applies for the other 2 reactions, where you can see that their reverse reactions will have positive delta G values. And to solve this problem, we will use phosphatase, which is the enzyme that will hydrolyze away the phosphate group. And with that, it will provide the energy to power up the reverse reactions. And now let's look at a bit more details. First, we want to convert the 3-carbon pyruvate into PEP, and in order to do that, we have to add a phosphate group onto pyruvate molecule. And we know that the reverse process is not going to happen due to the very positive delta G. So what we do is that we have 2 steps of reactions. In the first step, we are going to add a carbon to pyruvate by forming the 4-carbon oxaloacetate, and the energy will come from the hydrolysis of an ATP. And this reaction is catalyzed by an enzyme complex, which is pyruvate carboxylase, that will add a carbon dioxide onto the pyruvate with the help from the coenzyme biotin. But the problem is the oxaloacetate is found in the matrix of the mitochondria. Now, if you can remember, oxaloacetate is one of the metabolites in the Krebs cycle, which is in the matrix of the mitochondria. While the step 2 of this conversion will happen in the cytoplasm. So we need to transport the oxaloacetate out from the mitochondria. But the problem again is OAA is not permeable to the inner membrane of the mitochondria, and therefore it has to be converted into the other alternate form of metabolite. To do that, oxaloacetate is converted into malate by the enzyme malate dehydrogenase because malate is permeable to the mitochondrial membrane. And in the cytoplasm, then malate will eventually be converted back into oxaloacetate so that step 2 can then proceed. So the conversion of oxaloacetate into malate in the mitochondria is using malate dehydrogenase encoded by the mitochondrial DNA, while the conversion of malate back into oxaloacetate in the cytoplasm will be using malate dehydrogenase that is encoded by the nuclear DNA. Subsequently, the 4-carbon oxaloacetate is decarboxylated by PEP carboxykinase to produce the 3-carbon PEP. And this is another energy requiring step as we need the investment of GDP to provide the energy needed. And once we get into PEP, all these reactions can be easily reversed because they have a very low delta G values until fructose 1,6-bisphosphate To proceed further, we will need to release one phosphate group from carbon number 1 so that we can produce fructose 6-phosphate. As mentioned just now, it is impossible to have the reverse reaction because the delta G will be too positive. Alternately, we employ another enzyme, which is fructose 1,6-bisphosphatase that will cleave away the phosphate group from carbon number 1, forming fructose 6-phosphate. And it is the release of energy from this cleavage of phosphate group that will power up this reverse reaction. And then in the final reaction of gluconeogenesis, you have something very similar that we are not going to use enzyme kinase. Instead, we will use phosphatase, which is in this case a glucose 6-phosphatase, to remove the phosphate group from carbon number 6, forming up glucose molecule. With that, we have the synthesis of new glucose molecules in our liver, which is the factory of gluconeogenesis. And you can get pyruvate from different sources, which could be amino acids or lipids. And then the new glucose molecules can now be released into the bloodstream so that we can maintain our blood sugar level. And now let's talk about the energetics of gluconeogenesis. In glycolysis, one glucose can produce 2 ATP, while to produce one glucose through gluconeogenesis, we'll have to invest 4 ATP and 2 GTP, and that's equal to 6 ATP. So this tells us how important it is to maintain the blood sugar level so that both our brain and the red blood cell can function optimally. But the question is, where do we get the ATP? Now think about this. When we're trying to synthesize new glucose molecule, it means we are running out of sugars and carbohydrates. And therefore, this ATP must come from other alternate sources, such as the breakdown of lipids and the breakdown of amino acids. So we'll discuss about this in our final 2 lectures in biochemistry. In this paper, I just want to show you that if you are a trained athlete, your training is not just about increasing your muscle mass. It is also about regulating the enzymes. So if you have worked out for a long period, your body produces a signal to produce even more of the enzymes required for energy metabolism. So here's a piece of paper. What they did is that they tried to overexpress the enzyme PEP carboxykinase, which is the enzyme needed in the first step of gluconeogenesis, and something special happened. And let me show you through the video. You may now click on the video posted under Canvas, which is the super mouse video. So this has just proven that by upregulating one enzyme, our metabolism can be more efficient. But the big question is, can we do this to the people? Can we produce super athletes that can run forever? So these are the key findings from the experiment. First of all, it enhanced exercise capacity, which is 6 kilometers versus 200 meters. So that is equivalent to a 30 times increase. Imagine a full marathoner who can run 42 kilometers times 30. That would be more than 1,000 kilometers. And for the mutated mice, they ate 60% more than the controls, but only had half the body weight and 10% of the body fat. So this will be a dream come true for many people, especially girls and bodybuilders. And then it also increased the number of mitochondria, which help to utilize fats and triglycerides as the alternate source of energy. And last but not least, which is the most amazing part, that the mice had an extended lifespan, which is extra 2 years, as compared to the normal lifespan of 2 years. So 2 plus 2 is equivalent to a 100% increase in lifespan. This is just unbelievable. And if you could do this to people, we might have just solved the aging problem by tweaking just one enzyme. But to do this on people is really difficult, if not impossible, because this involves ethical issues. And if you boost up metabolism, we might also increase the production of ROS, which is the reactive oxygen species, that might have other side effects. So it is not as easy as you would imagine. So with these two metabolic pathways running in opposite direction, we do not want them to be switched on at the same time because they will cancel out one another. And therefore, we need a system of control. So when the cells require ATP production, glycolysis will be switched on, and they are activated by these activated molecules such as F26BP, AMP, and F16BP. On the other hand, when there is enough energy production, glycolysis will be switched off by the inhibitor molecules like ADP, citrate, and so on. So you can refer to the lecture with regards to the control of glycolysis. So when glycolysis is being switched off, gluconeogenesis is being switched on at the same time, and it's activated by citrate and acetyl coenzyme A. Why is it so? Because when you have citrate, it means that the Krebs cycle is already full, and when you have acetyl coenzyme A, it means that you have enough pyruvate because acetyl coenzyme A is converted from pyruvate molecule, which in other words, it means we do not need any ATP production while we have to conserve those molecules and use that to produce glucose molecule either for storage or either for topping up the blood sugar level. Now, on the other hand, gluconeogenesis can be inhibited by F26BP, AMP, and ADP. So, why do AMP and ADP inhibit gluconeogenesis, as you can see from the diagram? Well, because they mean a low energy status, and we need to send pyruvate into the Krebs cycle to get more energy production. Now, likewise, why do citrate and acetyl coenzyme A activate gluconeogenesis? That's because they indicate a high energy status, and it also means that we have plenty of biosynthetic precursors. So this favors the conversion of pyruvate into glucose and ultimately for the synthesis of glycogen for storage purposes. Aside from the allostatic control that we have just discussed, which involves the binding of activator and inhibitor molecules, we also have hormonal control, which is much slower, but they can control up to millions of cells at the same time. So in metabolism, there are two important hormones that you must understand, which is insulin and glucagon. So the action of insulin is to increase the expression of a glucose transporter. So with that, the muscle cells particularly can import glucose from the bloodstream. So that will reduce the blood sugar. And because you have enough glucose inside the cell, you do not really have to synthesize any more new glucose molecules. So insulin reduces gluconeogenesis. Now, on the other hand, if the blood sugar level is low, we will need the action from glucagon, which increases blood sugar level as it stimulates gluconeogenesis. So we need both hormones to ensure that our blood sugar level is being maintained and regulated. And with that, we have come to the end of this lecture. So please be informed that all the lectures in metabolism are interrelated. So in case you have forgotten the details from the previous lecture, you will always have to go back to those lectures and find out the details and try to link up all the different elements and components within metabolism.